Thanks so much a lot for the invitation, and actually it will be a bit less short. Sorry for that. So I want to talk about proteins, but I want to talk about it because it's a data science colloquium, so I want to talk about it from the point of view of data science. So my main work is actually now on biological problems and trying to understand how make the things work in relation to biology, but I will be relatively superficial on this part and more concentrate on aspects of modeling and so on with limitations. So since I want to talk about proteins, I have decided to make one slide introduction to proteins. Probably everything everybody in the room knows. So proteins are built by building blocks, which are the amino acids or residues. And there are 20 of them, and they have very different physical and chemical properties. And we will, in all the talk, completely forget about these properties and so on. But in some hand-waving arguments I will use, we will just have to remember that there are 20 different letters. And then proteins are linear chains of these amino acids, so they form linear chains, but these linear chains fold in a kind of hierarchical way, first into secondary structures, which form some well-defined three-dimensional folds, the so-called tertiary structure. Sometimes proteins come together to form complexes, several proteins, which is called quaternary structure. Proteins are super important in biology because they are basically involved in almost all biological functions. They have very, very many functions. I have just listed very few of them. It's not very important. But what is important is that most of this function is actually based on how these proteins fold exactly, how they interact exactly in a very precise way, because some part, for instance, will be so of the protein will be exposed at the surface, and at the surface it will be a catalyst for some reaction. So the fold is very important to understand the functionality, but what the situation in biology actually is that we have a lot of data at this level, and we have very, very limited data at this level. Because thanks to genome sequencing techniques, we have this can be done in high throughput, so we have many, many data. And here this is hard labor in the lab, so the data are limited. So one question is, can we actually use this data to learn something about this? That's obviously a data scientific question. I want to make this slightly more, illustrate this slightly better with this UniProt database, which is the most important protein database. So it's a database containing protein sequences and annotations of protein sequences. And it has two sub-databases. One is the size is characterized by the red line here, which you see has something like an roughly exponential increase over more than 1 decade. And this is proteins which come from genome sequencing projects. So the genome is sequenced. With some bioinformatic tools, you find genes, protein coding regions. You translate it on the computer into a protein, and you put the sequence into this database. So in principle, we have nothing but the sequence in this part. And you see this is growing very fastly because we have these very fast sequencing technologies. And here, if you ask what this little kink is here, at a certain point, the database was touching about 100 million entries. And people, so the curators became a bit nervous about the future of the database. And so they did a lot of work to reduce redundancy, to do it better. And they gained a factor 2. They wrote a very nice paper, which starts the title towards a more sustainable future, and so on. But then you see it took off again, and now it's at 200 million almost, 180 million proteins. So it's a lot compared to many, for instance, picture and Facebook or whatever, it's few, but it's already a lot of data. There's a second database, which is characterized by this flat blue line, which is those proteins where you have some manual annotations. Manual annotations means that biologists have been working on this. They have found out something about the function, something about the structure, something interesting about these proteins, which was worth to put in this data into this database. And so these are proteins where you have knowledge on, and this line is flat. It's really flat. It's almost no new proteins coming there. The annotations get better over time because people tend to work on the same proteins, and these are proteins which come what is called model organisms in biology, which is something like us, or which is E. coli, or which is the fruit fly and yeast and so on. So basically, there is more and more knowledge accumulating over a smaller and smaller subset of proteins. So what can we do? One important aspect of this data is that actually it's not just random sequences over 20 letters, but it is proteins. And proteins result from evolution. Since they result from evolution, you can organize them into what is called protein families, homologous protein families, and homologs are proteins of common evolutionary origin. This means these are two proteins, typically in two species, sometimes in one species, but two, let's say the simpler case is in two species, and they come from some protein which has already done the same thing in some ancestral species, which was the common ancestor of the two. So in the course of evolution, they diverge in sequence, and this sequence divergence is very important. So it goes as far as typically if you take two proteins from one protein, they have something like 20 to 30% of their amino acids in common, and 70–80% have been substituted in the course of evolution. So there's a lot of variability. But on the other hand, if you look to structure, if you look to function, to the aspects which were down in the first slide, they are very much conserved, even at these very long evolutionary distances. So you have a lot of sequences which are very variable and which do the same thing. So that's quite interesting. And also, it's important to say that if you take any of these sequences and you just introduce 10 or so random mutations, you can be almost sure that the protein is dead, that the protein is destabilized, that the protein is defunctionalized. So actually, this strong variability is very, very constrained by the fact that actually the structure and the function are still there. There's a database, like a lot, the so-called PFAM database, PFAM, like protein family, and which gives these families in form of so-called multiple sequence alignments, which look a bit like this one. So these are tables of these 20 letters and gaps, alignment gaps, which are needed to align sequences. So 21 letters, if you want to say. And each line here is one protein. Typically, the lines, so each has something like 50 to 500 positions, amino acid positions, variable according to the family. And but you have something like in good families of the order of 1,000 to 100,000, in some cases maybe 1 million sequences for one protein family. And all I do is modeling of one individual protein families. I can do this for many families, but I model them one by one. So alignment means that they are written in a way that they are as similar as possible. And if I go back to this figure here I have an overlay of two proteins which are actually at 30% sequence identity. And then an aligned position should be a position which corresponds to the amino acid in the orange and the amino acid in the blue one here. So should be the same position in the almost same fold. You see they have very similar folds. This should correspond to one column here. And if you stare on this table, you see it is not a random table. I have evidence, for instance, here, this column, which you see is all H histidines. And actually, it's not a case because the protein I'm showing here is called histidine kinase, and it's called histidine kinase because there is an H in this position. Other positions are quite variable, as you see here. So you see it's not a random matrix. It's something which contains information. And the question is, how can we get information out of this? So it's an intrinsically unsupervised task, what we have, because we want to stare on this and get something out of this statistical structure of the sequence variability. So I want to capture this sequence variability via some statistical models, which since I used to be a colleague of him, as he said, I always like to write as an exponential of minus some function which looks like an energy or a Hamiltonian, but that's just a convention in principle. And so I want to model my data, which is this multiple sequence alignment, via a statistical model. Obviously, one has to be careful about this, because my data, as I told you, are something like a few thousand sequences of length 50 to 500. And this model has, in principle, 20 to the length of the protein, so something like 10 to the 65, 10 to the 650 parameters. So I have to do some assumptions, some modeling assumptions about this model to do something reasonable. And what is done in the field in a quite standard way is to use the maximum entropy principle, so which is based on the selection of some observables, where observables are any functions from the sequence to real numbers. This can be anything what you want, and I will talk a lot about what one should take in terms of proteins, but it's in principle any function. So it can be the function, the question, is there a histidine in this yellow column which I showed you, yes or no? That's one function, but it can also be what is the overall electrostatic charge of the protein. It can be something which is very local, something which is very global. And then what I want to say is that my model should somehow reflect the empirical statistics of these observables. That's quite reasonable, and I don't even say the statistics, just I fix the average. So if I take my model P, which I want to model, and I'm sorry, I shouldn't have written a P here. This is not the same P as this one. But this is my model, my statistical model P. If I take the ensemble average of this one, it should match the sample average in my data. That's kind of fair. And beyond this, I want to have an S uniform model as possible, as unconstrained model as possible, so I maximize the entropy, and obviously I can do this. It's a very little constraint maximization problem. This gives an analytical form of my model in terms of the exponential of some linear combinations of these observables multiplied by some Lagrange multipliers. That's very simple, but obviously it shows us that there's a real problem, because my model depends explicitly on the observables I have selected. So I need some good criteria to select observables. So either I use some prior biological knowledge, which I will do in the first part, and then I will try to do some kind of objective criteria for selecting observables, but that's much less clear. Because I remind you, what we have in the first place is this table of 21 letters, or 20 letters and the minuses, and we have to model this one. So the biological knowledge I want to use is very simple. The first one, so what I want to insert is what is called conservation and coevolution or covariation. And let's first talk about conservation. This is exactly what I showed you with an example, with this histidine, for instance, which was totally conserved in the alignment because it was always there. Like here, I copied something in this totally fake alignment. I copied a column which is all histidine. And I would like to reproduce this. And this is encoded by what I will call Fi of A, which is the frequency of amino acid A in column I. So in this blue column, this would be 100% histidine and 0% all the others. Or in the green column, which is a variable one, this would be, I don't know, 1/3 R, 1/6 N, and so on. Okay? And I want to reproduce this by my model. But so most of sequence bioinformatics is actually based upon this observable on conservation and versus variability. And for instance, the alignment, which I showed you in the beginning, is constructed just using this information. Constructed in two ways. The selection of the proteins which are in a protein family is done using this kind of information, and the alignment, so to decide where, in which column to put which amino acid, is done using this information about conservation. So it's something which is maybe the most important information in sequence bioinformatics. But it's not all. There is something more in the data, and which is actually related to the fact that proteins fold into well-defined three-dimensional structure. So some positions which are distant along the 1D sequence, the primary sequence, fold reliably into contacts, so they touch each other, and touching means physical contact, so they should better have some compatible physical or chemical properties. For instance, putting two charged amino acids of equal charge close together in space, you know, is difficult due to electrostatic repulsion. So you should have, not all combinations of amino acids are equally well in different positions. And this is shown here in this very simple example that, for instance, R can be together with D, E can be together with K, but you have no RK and no ED. So now the second statistical observable I have is the co-occurrence of two amino acids, for instance, R and D, in two columns, which is this one. And you see that in the case, if these are correlated, then this Fijab is different from Fi of A times Fj of B. Like here, there is 50% Rd, and not 50% R times 50% D, which would be 25%. So you have some correlation in the occurrence of amino acids in some positions. So these are, I can write this for each column. I can write this for each pair of columns. And now I put this as my observables. And the maximum entropy model in this case gives me something which has this form, which is well known to many here, which you can call a Boltzmann machine, which you can call a Potts model, a Markov random field, depending on what is your original community. But the point is, it's an exponential model. And you have a linear combination of these coupling terms, which encode these ones, which are the Lagrange multiplier for these constraints, and this field terms, which are Lagrange multipliers for these single-site frequencies. Very simple. Obviously, here for the blue column, this would have a big field for histidine. OK. Now, how to learn? We still have, obviously, to learn the parameters to actually match the statistics of the model. So, for instance, the marginal statistic for pairs of amino acids in two positions should match this empirical Fij, which I just introduced. The basic idea is to do something like Boltzmann machine learning, where you start with some random initialization of all the parameters. You calculate marginal distributions. And then, according to the deviation of the marginal distributions from what they should be, so from the empirical observations, you update parameters. So if your Pij is smaller than Fij, you squeeze up a bit the parameter. Same thing, obviously, for the single-site marginals and the H's. So you can do this in principle. Only problem is that, obviously, the calculation of this object is a complicated calculation if you want to do this exactly, because you have, I told you, 50 to 500 letters here. Each one is has 21 possibilities. So this sum is a sum over 21 to the 50 to 500 sequences you cannot do, but you can do many approximations, some of them which are more based on statistical physics, like some more or less sophisticated mean field approaches, or you can do this just by sampling from your model by MCMC or by persistent contrastive divergence or other techniques. You can estimate this one and can try to fit your model. I don't want to go into any detail there. It can be done, and it's OK. I like very much this sentence by George Box, which was actually about statistical modeling of data, who says all models are wrong, but some are useful, because I postulated my observables and so on. So is this useful? Because I came up with a model which is relatively complex because I have all these interactions, so I cannot analytically calculate something like partition functions, marginals, and so on. Is this useful? And then how wrong is this model? And in this context of the Max and modeling, I want to rephrase this, are the pairwise couplings sufficient? So should I put more? Is it enough? Or are they really necessary? So should I maybe put less? And I know that what I'm going to talk about will make suffer those which are a bit more mathematically minded, because obviously if it's sufficient and necessary, I don't mean sufficient and necessary, but I talk about biology. So I mean something which maybe in a bit less stringent language would be useful, means that I can predict something, that I can interpret some of the parameters in a biological reasonable way. Sufficient means that the model is actually generative, so that sequences generated from my model, for instance, by Monte Carlo sampling, look like natural sequences, according to some criteria which I will come into. And necessary means that it's a minimum model or a parsimonious model, so using as few parameters as possible. I want to be very short on this first question, if models are useful. The answer is yes. If not, I wouldn't be here. And you can, there's a lot of information in the models. For instance, here on a protein, I have plotted the strongest couplings. So these Jij terms, the strongest one. And you see they are close to each other. They are not traversing all the protein. And this is actually something which has, important applications in protein structure prediction from sequence information. So it's just statistics which tells you how the protein is folded into. You can do this if you don't work with one single protein family, but with two protein families, to tell something about if these families have some protein-protein interaction, which is a very important topic in biology. Or, for instance, you can predict mutational effects, because, as I told you, for each sequence, we can now calculate the probability, or we can calculate the statistical energy, which is basically a log probability. And I can look, if I do a mutation in a sequence, if this probability goes up or down. So if it is tendentially a beneficial mutation or a deleterious mutation. And that's very important information, because you know that mutations are something very important. They can cause disease, they can cause cancer, they can cause antibiotic resistance. So they are important, but also they are important, for instance, in everything which is protein design, which is applications to improve, make engineering on proteins. You need to change the amino acid sequences, and you would like to do this in a rational way. So it is useful, yes, but there's a problem, because we have a lot of parameters. I introduced all these for all pairs of columns, these all pairs of amino acids. So this is something which, if you do the little calculation, it's something like 1 million to 100 million parameters, which is a lot, obviously. And that's why actually we have a lot of problems with interpretability. We have the biggest couplings we can interpret, but then there is a lot of parameters where we have no idea, no clue what they are important for. That's all about usefulness. So I want to talk a bit more about sufficiency, so about generative modeling. And now you could ask, what is a good criterion for generative modeling? And I would claim, since I'm talking about proteins, a good generative model, a good test would be actually to generate sequences, put them into some living being, some bacterium in my case, and see if it works or not. This would be a good test for generative. If it's generative because it gives me a real functional biological sequence, my model must be quite good. So the idea is to do sequence design. So very simple, starting from the multiple sequence of natural sequences, making this model, fitting this model as I told you, and then we can sample from this model. That's the easy part. The hard part is done by our colleagues, by our biological colleagues, which are experimentalists, Bill Russ and Rama Ranganathan in particular. We give them these sequences, so just a file containing the sequences, and they do experiments. And what they do is actually they take these sequences, they replace what is in a genome. Here, these are E. coli bacteria in my picture. So they replace in E. coli what is the real protein in E. coli, so the native protein in E. coli, and they replace it by something coding for either a different natural sequences, but which is not from E. coli, but from any other species, or by our generated sequences. Then they do some experiments. So they have some culture which is a mixed culture where different colors stand for bacteria which are genetically identically, but in this single gene which is coding either for the natural protein from a different species or for the non-natural artificial protein. We let it grow in some medium. They sequence the population before and after. So they count how much of this of the different proteins was here, how much of the different proteins was here. And then we simply calculate the number of sequences we found after time t relative to what was there at time 0, minus what the wild type, the natural sequence of E. coli would do. Take the logarithm. So this would be a positive number if actually this sequence here we are interested in had a stronger growth than the natural one, and it would be negative if it has less growth. Okay? What you find, and this is this relative enrichment here, so the protein we have been using to do this is so-called Chorismate mutase. And it is an enzyme. It's a biosynthetic enzyme which is responsible for the generation of 2 amino acids. And this is a very nice protein for experiments because if you grow the bacterium E. coli on a medium which contains these amino acids as food, it does not need to produce them, so it lives happily even if the protein is totally out, even if you just cut it out. But if you put E. coli, for instance, into sugar solution, so something where this amino acid is not present, it is essential. It will grow if and only if this protein, here, Chorismate mutase, produces enough of the precursor of these 2 amino acids, phenyl, alanine, and tyrosine. Okay? So this is very nice for experiments because you can keep you can multiply your culture and so on, but then you put it under the strong selection by giving it some food which does not contain the 2 amino acids, and it grows or not. So this relative enrichment I showed you, I was talking about, was first measured for the natural sequences. So it's a protein which has length almost 100. We had 1,259 sequences in the alignment of the natural sequences, which is a relatively small alignment, but was a good alignment which was curated by our biologist colleagues. So it's a nice alignment. And what you see if you put these natural sequences, so sequences which in their species are fully functional, if you put them into E. coli, there's no guarantee that they will function in E. coli, but you find something like a bimodal distribution. The coloring is just the cutoff, which I will use to say they are in this mode or in this mode. So there is a bimodal distribution of something which is close to 0, slightly negative, so growing a bit worse than what is the native in E. coli. That's normal. But many of them, 2/3 about of them, don't show growth. Actually, the experiment was also done on an E. coli where just the single gene was deleted. So again, identical, but there was nothing in this place. And this is pretty close to the maximum. It's just slightly below the maximum here. So this one is like none of this protein. This one is like working of this protein. Now, this is something which depends very much on the specific experimental conditions. For instance, if I overexpress a bit this protein, so I ask E. coli to produce more of this protein, actually this red peak will be more important, will grow, and this one will decrease. But these are the conditions which are used in the experiments on the non-natural sequences. So I have put down some numbers, some not very intelligent numbers, but which illustrate somehow about what are the sequence spaces we are talking about. So we have these lengths 96, and so all sequences of 20 amino acids of this length would be 10 to the 125 sequences. If now I would impose what typically computational biology imposes, which is this conservation, so what I called Fi of A. If I would just impose, then this position, there's a histidine, this position can be more variable, and so on. I would impose this. This would go down by about 40 orders of magnitude to 10 to the 86. Now, in my model, the entropy, or the number of sequences, so the exponential of the entropy goes to 10 to the 54. So again, another 30 orders of magnitude. If you compare this with numbers which are more physics numbers, then if you count the number of atoms in the universe, it's about 10 to the 80, which is a bit less than this one. If you count the atoms on Earth, it's 10 to the 50, which is a bit less on this one. And if you take one specific cell of your body, you have something like 10 to the 14 atoms in there. Now, if I say I take a totally random sequence and I ask it to be also coherent with coevolution, so to be a good sequence in my model, which is comparable to taking a random atom in the universe and asking it to be in one specific skin cell on my hand. Not very likely. If I already respect conservation, which is, as I told you, the most important signal used in sequence bioinformatics, this would be more or less, we would be closer to taking a random atom on the Earth and asking it to be in the same skin cell. So again, we should better have a good model, because we are talking about enormous sequences. So Kai is laughing, but the numbers are very simplistic, but I think they illustrate what they should illustrate. So is our model a generative model? The first thing it should be, obviously, we should fit well our model. We should do what we ask our model to do, which is reproducing the one residue, one point frequencies, and the 2-point frequencies, here connected correlations. And I plot the statistics of the natural sequences. So this would be the Fi against the statistics of an MCMC sample. So this would be the PI estimated, which I introduced before. And you see this is quite well fitted. We can do a bit better now, but this was a model which 3 years ago we gave to the experimentalists. But this is only fitting. So we did well our job. This one is about 2 million points here, so, and we have fitted them quite well. But we can also look to non-fitted quantities to convince ourselves that actually our model is something good. And here I put connected 3-point correlations. And there's no reason why the knowledge of 1- and 2-point correlations should tell me something about the 3-point correlations. It's very easy to show that you can have models like 3-spin models or so, which have some non-trivial structure on the level of the 3-point correlations and which are totally flat on 1- and 2-point correlations. But you see, actually, the natural sequences and the MCMC sequences have a very correlated signal. So this is something which is interesting. And there's something else which is shown here, which is this is just a simple principal component analysis of the natural sequence alignment. And you see it's a bit clustered somehow. Not very, but a bit clustered. And then I have matched I have projected my sample data onto the principal components of the natural alignment, and you see it occupies the same space, the same locations in sequence space. So it looks also good here, and here I have plotted again contacts. So this protein works as a homodimer, so there is a white one and the gray one, and you see the contact prediction is good. So my model seems to be a good model. So what does my model say about the sequences? First thing, the natural sequences. I have shown you the data. So what I have shown you before was just a histogram of this relative enrichment, so of these data. So you see the bimodal structure. And now I have plotted it against the statistical energy of my model I never used the name, we call this DCA, like direct coupling analysis. So we against the statistical energy, and you don't see anything, which is not surprising because we have fitted our model on all these data. So it should not discriminate between them. But we could not just fit on this one because the experiment was done after we have fitted. So we didn't have this information. Now we have sampled sequences from our model. And we have sampled sequence at the rather large range of the statistical energy, which is this log probability, making also some sequences which have very high energy, so would be very low probability. And you see, they never work. There is nothing in the relative enrichment in this red region. Whereas if you go to a range of the statistical energies, which is comparable to the natural ones, you see again this bimodal structure with a lot of functional. And these sequences are very, very variable. So they are not, there are many mutations away from any natural sequence. So this is quite surprising. And what is also interesting, if I would just not take this coevolution, so the model with the couplings Jij, but I would just use the model which has only the conservation, then my energies would be somewhere here, or maybe almost in the next floor, and none works, none. I don't show the data here. We have done the experiment. They don't work, and they should not work. So that's very interesting. This tells me that my model actually has some nice generative properties. There are some limitations which I'm putting a bit under the carpet. You can ask me if you want. Now, I told you that actually our model was fitted on all these data because we didn't have the information on the natural sequences. But now imagine that we would have the possibility to use some feedback. And in using feedback, I would say the simplest setting is that the experiments, if the experiment would have done the experiment on the natural sequences before, the one I showed you, and some of them are gray, they don't grow, and some of them are white, are red, and one, they grow. And I could have a very simple supervised problem to predict if this one is working or not, given the sequence. And we have done something which looks very similar to our Boltzmann things like here, which is basically logistic regression. We infer this from all the natural sequences and phenotypes. And then we look what happens actually with our sequences which we have sampled from the model, which I've shown you before. And this is what's coming out. So if you see, if you concentrate first on this low energy region, which I told you is basically the good sequences, you see that there is a very big concentration of gray here. And this one is the probability to rescue growth given the sequence. So if this is 0, my predictor would say the sequence does not work. If it's gray, it does not work in the experiment. Here I have a concentration of red. I would predict this works almost surely according to my model, and actually it works. Here I have 0.50, and you have a mixture of gray and red. So this looks quite well, but if you look here, there are also sequences here which my simple classifier would predict to work almost surely, and they don't work. But they don't work because they don't have this signature here, but they don't work because they have high statistical energies. So actually, these two things, this energy of my first model and this probability to rescue given the sequence, the supervised learning which I have, they capture two different informations. And I should have something like a logical end of them to actually have something which is really working. So to get back to my numbers, so these were the numbers where I started. This is what we got. That's a bit of propaganda, but sorry for this. We have, on all design sequences, we have almost 30% working. If we go to this low energy regime, which is given by the energy range of the natural sequences, we go up already to almost 40%. But now if I if I had the information before about what the natural sequences do, and I would have selected before only those which, according to my model, should have a good performance, I could have gone to 80%. And that's compared to 10 to the minus some big number. So I would say somehow, yes, it is somehow sufficient to have these pairwise couplings, because what I can do is I can sample from this model and these things work. They reproduce statistics which I did not fit. So it's somehow a good model, but it has a lot of problems. Again, there's a huge number of parameters, many of them not very well fittable. We need quite some regularization to actually fit the models, and this has some problems in actually sampling, and so there's a problem. I have too many parameters compared to the quite limited number of the amount of data, which was just 1,000 sequences. So can we do something instead of having all these little, very local observables? One position, is there a histidine, yes or not? Two positions, is R and K co-occurring in these two positions? Can I do something which is a more global, observable, something more collective, to reduce actually the number of sequences and the number of parameters? And the way we have chosen is the following. So we have imposed two types of constraints. The first one is actually the sequence profile, which we had also in the last modeling, and which is so important that we didn't dare not to put it. So we have put it. So these marginal single-site distributions reproduce what is empirically found in the data. So the statistics of the single columns. So this one we have kept, but then we have introduced something which is called sequence motifs in computational biology, and it's basically the simplest collective observable you can imagine, because it's an additive function over single sites, and you have a function of the single amino acid in this site, and you add this up. So it's something like, it's very similar, it's something which has very strong similarities, for instance, to principal component analysis, where eigenvectors have the same form somehow. Obviously, you have a different optimization problem because I want to do something generative, and principal component analysis is nothing which is a generative modeling. So we want to use this kind, and so we decide on a certain class of observables, and now we want to optimize over the choice of this class. So what are these omega really? So now we do this again by first doing this maximum entropy modeling, which I was talking about before. And obviously, I told you that for an observable, the empirical average should coincide with the model, the ensemble average in the model, which for this omega is nothing interesting, because the average over the multiple sequence alignment is just given by this average with this Fi. But since we fit them, it's always the case. So the average is already done. So we can go to the second moment. So let's do the second moment. And actually, the second moment contains now these co-occurrences, but in some combination, some non-trivial combination, not individually, but some combinations of them. Why should we care about the second moment? And I want to give you a very simple illustrative example why we do this. Imagine, for instance, my motif, my omega would be this one. We have just two letters, A and B, and we have three positions where the omega of A is positive +1, and the omega of B is negative -1. That's a very simple example. And remember this representation, which I will use on the real amino acids, and which will look a bit more messy, but it's basically the same representation. And now I have two different multiple sequence alignments, which always have 50% A and 50% B. But obviously, they are different because this one is more variable in terms of words, because this one has only two different words, and this one has four distinct words. And if you calculate, obviously, now the first moment of this omega here, it's 0, because it has as much +1s as -1s. But if you calculate the second moment, it's 5 here, and it's 9 here. It's bigger here. So actually, the second moment is not so so sensitive to the individual frequencies, residue frequencies, but it's sensitive to word frequencies. So actually, I find these motifs are over or underrepresented words, not just single amino acids, but combinations of amino acids. Okay, so if we put this into the Max-Ent modeling, for each of these second moments, I have to give one Lagrange multiplier. This would be my model. But still here, I assume that these sequence motifs are given. Now, the little step is, OK, I forget about that they are given. I want to optimize them. I want to find the best ones. So I want to find those motifs which maximize the likelihood of my data given the probability of my data given the sequence motifs. So we just introduce a new, well, we rescale these because this one is anyway arbitrary on a scale. So we rescale them, and what we optimize now is the same thing here, just we optimize instead of over the single prefactor, we optimize over all components of these motifs. And you see that this one is just the Hopfield model, or Hopfield-Potts model. The problem is we have to pay something because now having something which is an exponential over a quadratic form instead of something which is linear in the parameter makes our model non-convex. So we could have problems in inference. We actually don't have so much. We do this by doing a little standard trick of statistical physics, which is a Hubbard-Stratonovich transformation. So this quadratic term here gets linearized by some extra variable. And if you look, this one here looks exactly like the form of a restricted Boltzmann machine, where instead of, so where you have visible variables, which are our sequence, where you have some hidden variables, which are these auxiliary variables which we introduced in the Hubbard-Stratonovich transformation. So there are some hidden, some latent variables. And the coupling is only between latent variables and visible variables. So you have couplings. These motifs here become couplings between the two. And this is actually something, so it's a simple restricted Boltzmann machine because here we have just a quadratic potential on the hidden variables, which is simplifying our life because, or which is just coming out of the approach we used to generate it because we had this Hubbard-Stratonovich transformation. But you could do something, V of x mu, which is more complicated. And this is actually what Remy and Simona have done together with Jérôme here at the UNS. OK, now you can learn this. And one thing which is nice about restricted Boltzmann machines is that if you have given a sequence, these latent variables become conditionally independent. Whereas if you have given a hidden configuration, the sequences, the single positions become conditionally independent. And this is nice for sampling because you can start with some sequence. You can extract very easily a sample of the x given the sequence, and you can iterate forth and back, and that's a very nice way of iterating. And we have done this with persistent contrastive divergent, which is resumed here. What you find is that these Hopfield-Potts models seem to have quite good generative properties. I've shown you this kind of plots before. This is again for the one-point frequencies, so this is fitted. This is for the two-point correlations, which in this Boltzmann machine learning before were explicitly fitted. But remember, now they are not fitted because I have these sequence motifs which have some weird combinations of them. So already this one is something testing, and you see if we have very few patterns, just 5 motifs, 5 Hopfield patterns, it's not super well, but then it becomes successively better. And this is just the full Boltzmann machine. It's the best, but you come very close to it. And this one is 3-point connected correlations, and you see it's working also quite well. And for instance, here for 20 patterns, which is a case which we will discuss a bit more, we have only 1.8% of the parameters we had in the full model. And you see the statistics did not degrade so much. So it looks quite well. Now, talking about biology, I care a lot about interpretability. And if I look to the pattern, it's not this little AAA, BBB, but it looks like this. You have the entries for the different positions. You have entries which have certain numbers, and it looks quite messy. No, interpreting this one is not obvious. But you see, it's something which is a signal which is quite distributed. It's not just 1 or 2 or 3 sites. It's many sites. But it's also very heterogeneous. There are regions where you have a lot, and there are regions where you have nothing. So maybe you can interpret. So if we do this for more motifs, we observe that so this distributed but heterogeneous sequence motifs are quite typical. So this is all from the p equal to 20 case, where we have 1.8% of all parameters left. But what is interesting is if you actually project your natural sequences onto these motifs, you can calculate what is called an activity. It's basically our original observable. You see that some of them give a quite bimodal distribution. Some of them not, but many of them have somehow bimodal distribution. You can even convince yourself that these motifs are individually very important, because you can just look what happens if you remove one of these motifs from the model. So you just leave the other 19s. You sample from this model, and then you look, for instance, for the 2-point correlations if the empirical and the sampled from the reduced model still coincide, and you see it's quite messy. It's much, much worse than it used to be in the full model. But now the important point is that we have this bimodal distribution. And so now if we do something like in PCA, that we plot one against the other, you see that there's a clustered structure. You see, for instance, this is a full multiple sequence alignment. This is one of these motifs or patterns, and this is another one. And you see there is basically 3 clusters. But now this is a protein family. I know something about this protein family, and it has some functional subfamilies. So the proteins which are actually doing different stuff in different biological processes. And I know these annotations, so I just selected on top of complementary knowledge about these, I selected subfamilies. And you see there is this one is the biggest one, which is actually in the 3 examples I have always in this main blob here. But then, for instance, this one, this OMP-R class, in this component here, it has significantly larger values. It's basically giving this blob here, but not in this one. So one pattern is basically identifying, if it's bimodal, is identifying some small subfamily. There's always a smaller mode here, and that's actually the one I just showed you here. Oops, no. This one here. It's identifying subfamilies somehow. Now, this is also a key for interpreting them, actually. In this case, I know even more what these subfamilies do. And one thing they do is they form homodimers. So 2 times the same protein forms a complex. The individual protein of the different classes, I have 2 of these classes shown here, looks very similar. If you see this blue one here and this blue one here, they look almost identical. But now I have also shown where the second protein is located in the dimer, and you see it's totally different, the location. Because this blue one, if you look, is oriented in the same way, but one is here on this side and one is on top. And actually, the pattern, the motif identifying this class here has its concentration in here. And that's exactly the location of the interface. So you see, I have done this unsupervised learning. Some motif is coming out, and this motif tells me in this region there's something important happening. And in a case where I actually know something about the protein, I see that this important is real. Now, if I do this for a protein family where I know less, maybe this gives me some new information. Just as a very last point, There's a drawback of this model because all models are wrong, but some are useful. This is useful maybe for identifying subclasses and some structural interpretable motifs. But I told you in the very beginning that one of the main applications of this modeling is predicting contacts, so things which are distant on the sequence, but which are in contact in the protein fold. And this is represented here for this Boltzmann machine learning which I had before. And we have two histograms. One is all non-contacts, and one is all contacts. And you see now that the big couplings, so this is the size of the coupling, the strength of the couplings. It's a Frobenius norm of the couplings matrices. If it's big, it's almost always a contact. There are very few false positives here, but which are actually this interface contacts. So they are not really false positives. But if you now see, if you go to this Hopfield model or to the restricted Boltzmann machine, which has these distributed patterns, this tail disappears. For 20, which is what I showed you, this almost disappeared. So you lose this kind of information. So there's a trade-off, which I don't know how to handle, but which is kind of interesting. And we struggle to get both together somehow. So I come back to the slide, which you have seen several times. So about necessary, no, actually we can do much more parsimonious models. But it's very complicated because sometimes we look to a model which looks, if you just look to the statistics, very good. But then if you look, for instance, to the parameter interpretation, which was this contact prediction, it actually lost information. Again, remember that what we had in the beginning was this table, A, C, D, and so on, and the minus. And we wanted to extract information. And nobody tells us what the model is. We don't know what the generating model is. And it's complicated. Kept me busy for the last 12 years. OK, to close, I want to thank people in the group. Actually, I should update because Giancarlo and Anna went away, and others are joining. And I have evidence, in particular, the people who have most contributed to this part, which I've shown here, which is Kai on this restricted Boltzmann machines, which is Pierre and Matteo on the generative properties of the Boltzmann machine, and on generating the sequence for our experimental collaborators, Bill and Rama. And this was done in collaboration with Remy and Simona here from the UNS. Thank you.
Oh, thank you for the nice talk. How do you train the parameters? I mean, you mentioned Markov chain Monte Carlo, but just a metropolis.
It depends. So yeah, you can basically do some metropolis. Mm-hmm. Just you have to convince yourself that the data are decorrelated and so on. And details you ask better your neighbor. Sometimes, in some cases, we use something which does not care so much about equilibration, which is contrastive divergence or persistent contrastive divergence, which is illustrated here, which basically is for your learning, you initialize your sequences in the real data, which should be a good sample. And then you sample for a certain finite number of steps. And you update your parameters after this finite number of steps. And then you go on, you transfer just, you keep the data, and you update the parameters, and you continue your Markov chain basically as a non-homogeneous Markov chain. You update the model on the run. And there you don't care so much about equilibration, but if you think about this, when at a certain point the parameters are converged, then you basically have a very long Markov chain, mm-hmm. and you go to equilibrium. So actually the stationary point of this one should be the same one as if you would do, and it's faster. behind you, Luca.
Thanks for the nice talk. So if you take just one task of all of these, for example, protein generation, what are the different models, for example, deep learning models that could be applied to this type of task, and how does the performance you get from this more interpretable model compare with that?
That's a good question. Maybe we should talk about this in 2 years from now, because we just start to look into this. So what you need, obviously, is something which is a generative model. So this is already restricting. So people are looking into variational autoencoders in generative adversarial networks and so on. One thing, I'm not sure how much you can gain actually as compared to this one. So far, this is the only case basically where there is a lot of experimental data. Now maybe for GAN there was one preprint out, which came just out. One problem is that still you are in a regime where you have relatively limited data. So I would be a bit afraid that in many of these cases, these very flexible architectures of deep learning are actually just overfitting. But this is to be verified. So this is something which is, so let's say this one here is our first step into deep learning, where the depth is 1. So Remy always says shallow learning, and it is, but it already has some hidden units, come on. And it's an interesting question, because we postulate as the structure of a model. And in a more flexible deep learning model, we also postulate the structure, but it's much more flexible. So maybe we can have something which is in there which we don't capture. But comparing them is not obvious. And the experiments take 3 years. So it's not, we can't iterate easily. Postdocs stay 1 year or 2.
All right, another question? Or we have to wait 3 years to get the answer for this one? So.
To wait for the question.
Of course.
So the other question is there also some different type of data that you could use to train these models? Like all these data seem to be static in some way. Is there a way of obtaining, I don't know, some different time frames of a protein, some evolution of a single protein, so different type of structured data?
Yes. Yes. This is addressing, again, a super hard question, because as I wrote it to you, it seemed as if you would assume that these protein sequences would be an IID sample, so that basically evolution would be an IID sampler from some Potts model, which obviously is totally wrong. Sequences are very much correlated. There is some hierarchical structure given by the phylogenetic tree. And what we do for the moment is something very stupid to play with. This is basically we give different weights to sequences. And if we have sequences which are too close to each other, we give them a lower weight, because basically it's almost the same. This is helping a bit. But putting the two things together, actually doing something which is like a dynamical generative model, is very complicated, because in a setting like these models here, you have to calculate, you have to characterize a propagator to go from one sequence to another sequence in a certain time. And that's a super hard object. So that's something where we are fighting, and it's tough. On the contrary, on what is, for instance, the contact prediction doesn't seem to be so important. And what is typically done now in sequence bioinformatics, due to the fact that sequences are exploding, so that there's more and more sequences, just to do some downsampling, to kind of redistribute your sequences in a more IID-looking way, whatever this means.
All right, if there are no more questions, I think it's a good time to thank Martin.